Sia, thank you so much. And many thanks also to Kirsty Jerrigan for helping so much to make this dream of ours come true to get Sia to London. It's my great pleasure now to introduce our next speaker, Tim Bliss. It was quite at the beginning of the research to the marathon that Julia rang me one morning very early and said she had an extraordinary encounter and it would be an absolute complete emergency that we would meet Tim Bliss. And very soon after, Nicola Lies, Lucia Pietro Justi and I went to see Tim. He's become one of the key advisors to this marathon, so we're very grateful to him. Tim was born in England and gained his PhD at McGill University in Canada. In '67, he joined the MRC National Institute for Medical Research in Mill Hill in London, where he was head of division of neurophysiology until 2006. His work with Terje Lomo and Per Andersen's lab at the University of Oslo in the late 60s established the pioneering phenomenon of long-term potentiation, LTP, as the dominant synaptic model of how the mammalian brain stores memories. Tim is a fellow of the Royal Society. He also has authored many books and gave recently the annual Coronian lecture actually in May 2012. Today for the marathon, Tim will be presenting a talk called The Machinery of Memory. A very warm welcome to Tim Bliss.
Good morning. I'm a scientist thrown among artists, so be nice to me. I'm going to talk about memory in its most basic sense and what actually happens in the brain when we store memories and when we retrieve memories. So it's a 15-minute talk on the neuroscience of memory. So we begin with, and I'm afraid I've got slides, and I don't know how well you're going to see them, so I'm going to sort of describe them as I go along. So, and apparently the arrow here, there we go. Can you hear me? This sounds a bit... So what we have here is, okay. That is, yes.
Bring these forward.
Oh, yes. Well, if you can see this. Can we have these? Please. What's shown on this first slide, this is a bit of history. The modern era of neuroscience began about 100 years ago with the great Spanish neuroanatomist Ramon y Cajal, and here is Ramon y Cajal in his lab in Madrid. And he established what's called the neuron doctrine, which is that basically the nerve cells in the brain receive information on their dendrites, structures called dendrites, and they emit information. The nerve impulse travels down an axon, and so you have information coming into the dendrites and then down the axon. And he also discovered structures called synaptic spines. And that's part of the machinery of the synapse, and the synapse is the junction between one nerve cell and another. So the most important structure in the brain from the point of view of memory is in fact the synapse, because that's where changes occur that store memories. That's what we believe, it's what Cajal believed, and it's what we believe today, and we are some way further than Cajal was, but we're not quite there yet. So synapses have what's called a postsynaptic side and a presynaptic side, and there's a gap between. So when one nerve cell connects with another, it does so across a little gap called the synaptic cleft. Okay. So there is a picture of spines, which we won't bother about because I don't think you can see it. Now here's a diagram of the structure of a synapse. All right? So we have the presynaptic part, which is filled with these little circles, and they represent vesicles, globular structures, which are filled with transmitter. The thing about nervous transmission is that it's electrical until it comes to the synapse, and at that point it becomes chemical. So the act of nervous transmission occurs when a nerve impulse reaches this presynaptic site, and at that point, one of those little round things fuses with the membrane and releases some transmitter substance. For an excitatory synapse, that transmitter substance is called glutamate. And then it binds, the released glutamate binds to specialized proteins which are embedded in the spine. That's on the postsynaptic side, the target side. So you've got a presynaptic side, a synaptic cleft, and a postsynaptic side, and you get the release of the transmitter binds to receptors, and that either excites or inhibits the target cell. That's the process of synaptic transmission. Now, when you think about memory, it seemed obvious from the very beginning, the time began with this subject 100 years ago, that probably information was stored at synapses. And that became even clearer when it was established by Lord Adrian in Cambridge in the 1920s, that the nerve impulse itself, the so-called action potential, is an all or none event. A nerve cell either fires an action potential or it doesn't. It reaches a threshold, then it fires, and that nerve impulse, that electrical impulse, travels down the axon to the next cell. And that's all or none. You can't change that. So it's unlikely that a memory is stored as changes in the size of nerve impulses. That's not going to work because they don't change their size. They're all or none. So what's left? What's left is the synapses. So a Canadian psychologist called Donald Hebb thought about this in the 1950s, and he came up with what he called a neurophysiological postulate. Hebb's neurophysiological postulate is dead simple. It says, consider a nerve cell, let's call it B, and consider one of the nerve cells that are projecting to nerve cell B, let's call that A. So A projects to B. It's not the only cell projecting to B, but it's one of them. And Hebb simply said, well, if A and B are involved in some sort of memory circuit, then let's suppose that whenever A fires and helps B to fire, that will make a stronger synapse. If that were to happen, you can see that, that a memory or that an event, which of course is encoded by the brain simply as a pattern of neural activity, so it has to, if you're going to store information in brains, it's got to be the activity of brains that change brains. And what Hebb said was, well, if cell A helps the cell fire B, helps to fire cell B, that synapse will become stronger, simply as a result of that activity. A very simple idea, and, but one that's been enormously influential, and one which is actually turns out to be true, we think. So, where can you find Hebb synapses? In 1950, there weren't any synapses that had that property known in the mammalian nervous system. So there was a search to look for places where you might find such synapses. And the hippocampus seemed a good place to look, because the hippocampus is structured in here. And if you want to lose your memory, Jani, get rid of your hippocampus. Don't have a prefrontal lobotomy. Because if you lose your hippocampus, you will have no more ability to form new memories. Memories, everyday memories of your life. As we know from famous amnesics like H.M., a young American who in the 1950s suffered from terrible epilepsy. They couldn't control it by drugs, so the surgeon had the brilliant idea of removing the structure that was giving rise to his epileptic fits, and that was the hippocampus. And from that day to the day he died a few years ago, H.M. was not able to form any new memories. His memory span was about 5 minutes, and then it was gone. So clearly the hippocampus is involved in memory. It also turns out that the hippocampus is a rather simple structure in neural terms. The neocortex is frightfully complicated compared to the hippocampus, which is rather simply organized. And that makes it easy to do experiments. And of course, science, as Peter Meuron says, is the process of scientific research, is the science of what's possible. So it was possible to do the kind of experiments you needed to show whether or not there were Hebb synapses in the hippocampus, but it didn't help that we knew that the hippocampus was an important structure for memory. So here's a diagram of the hippocampus with some electrodes which are going to give us information about the strength of that process of synaptic transmission. And it so happens that because, for reasons I can't go into, but it has to do with the simplicity of the organization of the hippocampus, it turns out that you can do experiments for a very long time. And for instance, you can implant electrodes under anesthesia, of course, into a rat, and then allow the animal to recover, and then you can do experiments with these electrodes, which will measure the strength of transmission, synaptic transmission, for large amounts of time, days, weeks even. And that's what you want to know. But what you're looking for here is synapses which can change their efficiency for long periods of time, preferably indefinitely, because we know that memories can last indefinitely. So here is an example of the kind of experiment which established that there were Hebbian synapses in the brain because you could give brief trains of intense activity across these particular synapses, and they would become better synapses. They would become more efficient almost immediately, within a few seconds, and then they would stay at that elevated level of efficiency for as long as one could do the recording. So here was a phenomenon which looked like it behaved as a Hebb synapse. The question was, did the brain actually use this phenomenon to store memories? Well, that turned out to be actually a much more difficult thing to establish, and still isn't completely established. Although if you took a poll of neuroscientists, I guess about 98% would say that memory involves these Hebbian synapses. I think this is a rather good experiment, and I think I'm just going to describe this briefly to you because I want you to leave this pavilion being able to have an intelligent conversation with the next neuroscientist you meet. So this is neuroscience for bluffers. Now, it turns out that yeah, here's this diagram again. It turns out that there are two kinds of receptors on the postsynaptic side, that's on the target side of a synapse. And one of those receptors, it's called the NMDA receptor, okay? That is one of the most important receptors, one of the most important proteins in the whole of neuroscience. It's probably involved in all sorts of things which make the brain not work well, like schizophrenia. But it's also very important for memory, for this model of memory, long-term potentiation, because it's what controls whether or not that synapse gets potentiated. And it has some peculiar properties which mean that LTP, long-term potentiation, has not just the property that it lasts a very long time, which of course is a sine qua non, but also that it has what's called input specificity. So in the limit, you can imagine a single synapse on a cell, and a typical cell in the brain will have thousands of synapses. A typical one can be individually potentiated. So it's not a cell-wide phenomenon. It can be restricted to individual synapses, and that greatly increases the memorial power of this arrangement. I mean, if each individual cell can be, each individual synapse can be tweaked, then you've got a much more powerful memory system. What I want to show here is, yes, you see, what we have here is something which I showed to Richard Wentworth several years ago now, and he was rather entranced by. And I think I'm still entranced by it, because what we're actually seeing here are synapses in action. Here, the structure here is a dendrite, and that little thing there is a spine. And you can see that it's flashing. And that spine is flashing because it's received transmitter. That is an actual act of synaptic transmission in more or less real time. And so we've got techniques now which allow us to look at the act of synaptic transmission and to look at the mechanisms that support long-term potentiation. And basically, there are two ways in which it can be done. A synapse has two sides. And what we're talking about is an increase in synaptic efficiency. How can that synapse become more efficient? Well, it could either be better at releasing transmitter, or it could release the same amount of transmitter, and yet the proteins on which are embedded in the postsynaptic membrane could become more efficient, or more proteins could be trafficked into the membrane. Two ways, what's called a presynaptic way or a postsynaptic way of making a stronger synapse. And the interesting thing actually about the presynaptic way is that it's not a case of actually releasing more transmitter. It's a case of making that process more reliable. Because the process of synaptic transmission is not 100% reliable. A nerve impulse will invade the nerve terminal, and it may or may not release transmitter. It's a finely balanced process, and sometimes it works and sometimes it doesn't. So if you could fix things so that it became more efficient, that would make a more reliable synapse, and that's another way of encoding memory, if you like, at the synaptic level. Or you could take the other side and say, well, let's just shove in more receptors, and that'll make a stronger synapse, which indeed it does. And there's evidence for both of these ways of making a stronger synapse. So to finish, I want to ask some questions about this phenomenon of LTP. We know that we can make stronger synapses. These synapses have this Hebbian property. Well, do they actually encode memory? And that is a much harder job. We know a lot about these receptors and the process of synaptic transmission and so on, because that's fairly easy to study. But behavior is not easy. And relating cells and networks of cells, vastly complex networks of cells, to behavior is also not easy. But you can start off by saying, well, what sorts of things would you expect to happen if LTP, this example of a Hebbian synapse, were to be responsible for encoding memory? Well, you would say, what happens if you block LTP? And there are drugs which will do that. You can block the NMDA receptor and you won't get LTP. What does that do to memory? So in experimental animals like the rat, the hippocampus is really important for spatial memory, and rats are really good at finding their way around. So you can train them to find their way around a maze or something. And then you can apply a drug, a drug which blocks the NMDA receptor. So the long-term potentiation can't occur in those animals, and ask whether that actually prevents them from being able to learn. And by and large, it does. But, you know, it's by and large. There are a few exceptions. Does that make everything, I mean, does that prove that long-term potentiation is not the neural basis of memory? Well, no, it doesn't, because there are lots of ways in which you can escape that conclusion when you're dealing with trying to relate a physiological phenomenon to a complex behavior. We're only just beginning to acquire the tools, in fact, that will allow us to study neural networks, large numbers of cells in the living animal. So my list of things that you should be able to do if LTP has got something to memory, to do with memory, by and large, it works out. But there's one here which I'd just like to briefly mention, and that is, if we're right about this, if memories are a distribution of changes in synaptic strength across certain networks in the brain, the more we know about this, the nearer we will come to the day when you'll be able to insert memories. Even now, we think we have pretty good ways of dampening memories, abolishing memories, which can be clinically useful. But we don't have a way of saying, you would like a memory of that night you spent with Marilyn Monroe. Well, I can fix that for you, for a price. We're not quite there yet, but in principle, we should be able to do that. Because if that's what memories are, and we have the skill and the techniques to get to individual synapses, and that might come one day, but I must say that's not quite there yet. So, in conclusion then, let me say that
the neural basis of memory is certainly, nobody really disputes this, it's certainly activity-driven. That's to say, it's the activity of the nervous cell itself which changes its own components. It changes the strength of its own synapses. And that's, at the physiological level, that is what memory is. At the psychological level, at the behavioral level, there's really a colossal gap between our understanding, what we understand about the cell biology of changes in synaptic strength, and how this is encoded by large numbers of synapses. So thank you very much. I would like to leave you with just one takeaway message. There are two sorts of memory: short-term memory, long-term memory, and short-term memory. Thank you very much.